nice to see all of you. And is my presentation visible right now?
Yes,
Okay,
we can see
great.
your screen.
And well, where I have here. Okay. Right. So yes, I'm taking you not just for the north, but it's almost like 500 kilometers north from the Arctic Cycle in the Yana Indigirka lowland in Siberia. We worked in a multi-author team on the Alas Lake profile. For that who don't know what is Alas Lakes, it's a kind of drying out lake in a shallow thermokarst depression. It's very interesting type of ecosystem. And as you can see, there are plenty of them in the studied lakeland. We are right here in the Indigirka Valley next to the Belerek River. And as you can see, there are two Alas levels, and we are studying the sediments, first of all, the sediments from the lower Alas level. The upper one is waiting for a better time, let's say. And what we wanted to study, we wanted to reconstruct the habitat changes and evolution of that lake, identify the hydrological changes and their causes, tracking climatic fluctuations, and expand the knowledge in understanding the response of Arctic ecosystem to climate and environmental change. So about chronology, we have in our short core hiatus, somewhere in the middle, as you can see. We have bottom part dated to, let's say, 3,000 years CALVP, and the uppermost layer with no contemporary deposits, which is confirmed by the lead analysis. The sediment character is two-folded, and we can put the sharp line from the clayal silt, then the hiatus and such pit, which is already visible in the geochemical analysis. But when we look deeper, we will see two more phases, transition one between the previous two, and the topmost layer with higher mercury concentrations, which is quite interesting. But what I want to show you, the insect remains which were recorded in the deposits. I'm first of all focused on chironomids, so there were quite a plenty of them. We were quite surprised by the amount. And in the first phase, there were dominating and quite high share of cold stenotherms, characteristic also to oligotrophic water. As you can see, there was also some pollen of, for example, Dryas octopetala, which were confirming such record. But along this taxa, there were present also many eutrophic and mesotrophic taxa, as well in chironomid stratigraphy and among plants. So there were green algae, karate, and so on, but also some other aquatic organisms, aquatic animals like cladocerians. But the next phase is defined by the presence, higher presence of semi-terrestrial taxa, semi-terrestrial chironomids, and coprophilous fungi, which were indicating probably pastures for large herbivores. It's because our ecosystem transformed from thermokarst lakes to a kind of grassland. And constant terms decline, so we can suppose that the change of in ecosystem character was caused by climatic impulse. The third phase is very, very interesting just because it's already a seasonal water body. So we can see here taxa like Trisocladius among chironomids and Branchinecta paludosa among macrofossils, which indicate drying out in summer and freezing in winter. So it's definition of that water body in that time. It was really shallow, really overgrown, as indicated by phytophilous chironomids. Very, very interesting. And the last phase, it was similar to the second one. Also some oribatids, some chironomids, semi-terrestrial chironomids. Also another interesting subfossils. But here you can see also a Rheocricotopus, which is Rheophilus chironomid, which can indicate some phases of drying and massive floodings, which we were told that are typical to that landscape right now. And what we do is water level reconstruction. We have, you can see at the water level curve that first of all we have shallow but lake, typical aquatic conditions. Then we have sharp decline of water level depth and some fluctuations in the phase of seasonal water body, and then some increase in water table, which might be connected with the flooding events. And it is, we suppose it is interconnected. The dry phases are interconnected with drying indicated by the tree rings from the paper of Turakova, Sidorova et al. We can see that it is quite consistent. And of course, we can see also that in these dry phases, there is a decline of Betulanana in favor of Salix and then also Larix, and also a decline in Cyperaceae share in favor of Poaceae, which, as I told, were kind of pastures for large herbivores in that time. I don't know how much time do I have more, but I would like to also mark a nice share of Trichoptera remains, which my colleague wanted to talk about, but she cannot be here right now. So just to mark that it was really, really nice analysis with some preliminary results. And just to conclude, that the river activity may cause lake drainage, leading to habitat shift and sediment hiatus. Probably the other cause coexisting cause was climatic impact. We plan analysis of 2 more really short cores. Of course, we plan also for the radiocarbon dating and geochemical analysis. But what I like to say that consistent reaction of many analyzed organisms. We analyzed chironomids, other insects, plants, geochemistry, everything, also diatoms and cladocerans. Every organism we analyzed had answer to environmental changes which were consistent, which proves the high sensitivity of Arctic ecosystems to environmental changes. And the insect subfossils serve not only as a bioarchive of the ALAS evolution, but also past fluctuations in water level and interconnections between the elements of the ecosystem, which are planned to be further studied. Thank you very much. I'm ready for questions.
Thanks so much, Olga. Dziękuję. Amazing talk. We all love multiproxy reconstruction, so it's great to see the first presentation in this meeting. Thank you. Questions for Olga? Any chironomid guy in the room? Not me. But I may have some questions, of course. But
also some nice macrofossils, both plants and animal macrofossils. The really nice was the resting eggs of Anostraka, Brachynecta paludosa, which was recorded for the first time from the sediment core here. It's not yet published. But also the resting eggs of Hydra, which were interconnected with Anostraka. That was really interesting.
We have a question in the chat, actually. Oh, yeah, some. From Sreja is asking, how do we reconstruct water level from insect subspecies?
Subspecies. We are not working on subspecies, but morphotypes. So we have we are using transfer functions, which is a method of taking data from the modern data sets and transferring the results, the ecological optimas, to the fossil ones. I'm not sure if that was the question, but yeah.
Yes, so we have another question, actually. I don't know if we have time.
Yeah, I think, we still have a couple of minutes or minute and a half. So Luciana is asking, considering the multiproxy approach you adopted, she would like to ask, what were the main challenges you faced in disentangle ecological signals when working with distinct biological proxies, such as chironomids and ostracods, and how did you address potential mismatches in their temporal or functional responses to environmental change? So that's a big question, Olga, if you want to try to answer it.
Yeah. As I said, the responses of almost every organism were quite constant. So we were quite surprised with that. It were providing like a full picture of that ecosystem. The problems we faced were, for example, in the lack of diatoms in the lacustrine phase. it's probably the preservation case. But besides that, maybe I haven't talked about it more, but everything was quite really consistent, and we are quite shocked with that. For example, where do I have it? Yeah, here. The share of planktonic to littoral cladocera, you can see it's very, very consistent with water debris constructions on the basis of chironomid head capsule. So both animals, plants, and so on and so forth were very, very consistent in the reaction of environmental changes.
Can I ask one question?
Yes, Muhammad.
Yeah, I have a question about that. What is the source of that microfossils which are present in your study?
We are taking, maybe I haven't shown the photo, we are taking a sediment core which were taken directly from the ALAS, right? So it was taken by Instorf sampler, directly from the ALAS lake, or not lake right now, yeah. ALAS ecosystem.
Okay.
Yes.
Okay.
Yeah, that makes sense. Thanks. Yeah. I think it's a really cool ecosystem to study, because I can see lots of interactions between the aquatic and terrestrial phases.
Yeah.
So even if it's so dynamic in time,
yeah, it is.
The thing that you see a very coherent response across so different organisms makes not only the ecosystem very responsive to climate change, but also the organisms themselves. So yeah, that's
yes.
So it's very interesting.
Exactly. The response of organisms was instant. So it was really nice to see it. Yes.
Great. Thanks, Olga. Thanks so much for the talk.
Thank you very much.
Amazing. So with Olga's talk, we reached the end of the first part of this first session, pushing the boundaries. So now we have time for a little break. But before that, before people start, disappearing, Liz wanna do a short announcement.
Wake up, everyone! So this is for waking up. Stand up, make a. So we're gonna make a picture. Thank you, Angie, for doing the design. So we want to capture this moment together. Please turn on your camera if you can, and don't worry, you're gonna look great. Remember that we're gonna look like in the posts or whenever, like very small. So Angie and Edward are helping us doing the screenshots. So I don't know, Edward and Angie, you just let us know so that we try to smile. Don't close your eyes.
Okay, I'm ready. So smile.
Smile, everyone. Thank you for being here. Oh, it's really nice to see some faces. Also from my lab. Thank you for being there. So you let us know, Edward. Otherwise, it's hurting. You have everyone?
Yes.
Yes?
Yes.
All right. So some people also moving. Very nice. All right, everyone. So we're going to go now to the coffee break, and we will return in some minutes. Be at 6 Central European Summer Time. Some 5 minutes before, because we're going to start at 6 p.m. with Jonathan. So see you in a bit. Bye-bye.